offers his own unique perspective of how to integrate this information into the practice of medicine. Is Issac Kohane, is the director of the Children's Hospital Informatics Program and is in the Henderson Professor of Pediatrics and Health Sciences and Technology at Harvard Medical School. He's also co-director of the HMS Center for Biomedical Informatics and director of the HMS Countway Library of Medicine, which in my opinion is perhaps one of the greatest biomedical libraries amongst other resources in the world. So.
Alrighty. Well, good morning, and thank you for having invited me. I think that you'll find this talk is both a complement and a counterpoint to what you just heard from Mike Snyder. And I'm going to talk to you about this mysterious thing called the incidentalome, which, let me tell you, is going to be really, really big. Here are the disclosures. I'm on the scientific advisory board of these two companies without equity. And I have to also say, as an observant Jew, looking at the Sabbath elevator and various signs around here, I actually felt more home than home in this hospital. So one of my former students is Atul Butte. Atul Butte was MD PhD with me, and he did exactly what Mike just told you. He did this marvelous report where each one of those triangles represents, for that disease, what is the population prevalence for that disease, and then the little lines, red or green, are measures of how much does your genome change that risk based on the reading of the literature. And with all the problems that we're about to talk about, I still think it's a remarkable document, but I think that we have to be careful about what we think about, how we think about it. And I just want to remind you, behind all of this is a pipeline. There's a pipeline that goes from measurement with sample prep and sequencing that requires assembly from all the short reads to create all the variant calls relative to which reference genome, and to summarize what's known, rightly or wrongly, in literature, and to summarize this at the variant level, at the gene level, and the pathway level, and ultimately at the level of a whole human report. I want to point out that what Mike showed us was there was no report. It was a bunch of really smart guys and girls looking at it and saying, this might be interesting, that might be interesting. There was no patient, doctor-readable report said, this is what you can take home and bank on. And so the challenge, you hear a lot in the, my PhD is in computer science, and so in some of the, and by the way, I'm a pediatric endocrinologist by training, what you hear a lot on my PhD side is how these omic personalized medicine challenges are ones of computational speed, of the size of the data pipes and the size of data storage. Those are transients. I don't think we have to worry about it. What we do have to worry about is this. There was a very interesting proteomic test, and the evidence for it was better than anything you have heard about today. Nothing you will hear today had better evidence of this in asymptomatic individuals. And on the basis of this proteomic test, hundreds, millions of individuals with a Y chromosome, with an effective Y chromosome, had individuals with very sharp knives come and take out this organ from between their legs. That, of course, is the prostate-specific antigen, and that is the prostate, which is now missing in millions of men. At great cost of billions of dollars to the healthcare system, at great cost to lifestyle, sexual function, at great cost personally of just lifestyle, urinary incontinence. And it was very, very foreseeable. I used to give this lecture, this slide rather, 10 years ago, and I said, how come there's 3,000 articles about the right way to interpret it still today? That was a very personal profile. And guess what? We finally, decades later, having caused harm to untold hundreds of thousands, if not millions of individuals, we're taking it off as a recommendation for most men. I think it's very sobering when we think about what we're going to do in personalized profiles. It's not just nice to know. Things happen when you're worried about the future, and things happen even more clearly if there are individuals who happen to have MDs who are telling you that you should worry about it. This led me to coin a term, the incidentalome, the biggest ome of them all, bigger than the genome, transcriptome, proteome, metabolome, all wrapped in together, and it's a growth industry. I was so enamored by this term that I went to Wikipedia and entered it into Wikipedia. 2 weeks later, I ego-surfed back to Wikipedia to see my entry. It was gone. It's a wiki. It has a version history. So I looked at the versions and I saw it was deleted by an individual. And I sent him or her an email. I said, "Why did you delete my entry?" And he or she politely said, "It's a very interesting concept, but there's no authoritative reference." So I said to myself, "Be that way." And then I went with my friends Dan Macy's and Russ Altman at Stanford and Vanderbilt and wrote a paper called "The Incidentalome: A Threat to Genomic Medicine," and got it published in the Journal of the American Medical Association. Then I went back to Wikipedia, re-entered it, referenced my JAMA paper, and it's stuck since then. So there's an interesting comment there about new versus old media. However, there is substance behind this idea. If I gave you the world's, let me teach you something new in PowerPoint. If you hit the B key, the screen goes black. If I gave you the world's most powerful, most exact genomic test, so good that you would run out of this conference room right now, despite the rudeness, and go and call up your friendly VC. This genomic test I'm giving you is 100% sensitive. It is 99.99% specific. And for those of you who don't deal with these kinds of numbers, you should know most genetic tests are about 80, on a good day, 80 to 70, 80% specific and sensitive. But I'm giving you this wonderful test. And now, instead of doing one test on you, I do not 2,000 tests, I don't do 4,000, I do 10,000 such tests. And by the way, for $200, I can now do 1 million such tests with a SNP chip. What this tells you is that if each test has those remarkable characteristics that exceed anything that Mike Snyder has told you about, 60% of the United States population is falsely positive. 60%. And what would that mean? It would mean not only do we have a bunch of angry, worried patients, like when my wife had a mammogram and we saw something we had to worry. Okay, we don't care about patients. How about a bunch of doctors that are getting phone calls from patients saying, I need a follow-up test because I found this risk. Okay, so doctors are angry. Who cares? But what then happens when the insurance companies start getting a deluge of tests, follow-up tests, not the genomic test, but the follow-up test for something that was not real? You know what happens then? Not only will they not pay for the genomic test, which they already don't, they won't pay for the follow-up tests. That would be the end of genomic medicine. And so the point I made in this article is we've got to clean up our act and figure out when is the right time to order the test and how to interpret it before we go crazy about it. When I first presented this article, I think I came off, and maybe accurately, as a bit of a crank. But then there was this article that appeared in Nature, Genetics perhaps, by a group of people who had sent in, I think Craig Venter was one of them, 5 of their spit samples to, I think it was 23andMe and Navigenics. And they got their whole genome profile. And look here what we have. Everywhere you see a tan box, not only is the size of the risk different, according to these 2 platforms, commercially vetted and with some degree of FDA approval, they're in different directions. different directions. Not good. I got further worried about it when I saw an article in 2008 about this guy. They had just resequenced, I think 454 sequenced his whole genome. And this appeared in Nature. And in table 3 of that paper, he was found to be homozygous for 3 diseases that in the books are highly penetrant, Mendelian inherited diseases that are present in childhood, like cocaine syndrome, for example. And although this guy, James Watson, is a weird guy, he's not that weird. And when I looked at fine print why they thought he was homozygous, they sort of in a mealy-mouthed way attributed it to sequencing error. But I was thinking, no, this is not sequencing error. This is the incidentalome coming to roost, and it's about to grow. And then I got even more courage when I saw this very nice study that just appeared in Science, Systematic Survey of Loss of Function Variants in Human Protein Coding Genes. And when they looked at a number of whole genome sequences, and they found loss of function alleles, major termination, frame shift mutations in a number of genes that apparently did not cause disease. And furthermore, they showed with biological validation that 20 out of the 100 that they looked at were fully inactivated. Now, those of us who have lived through the 80s of knockout mice, when we saw every time we knocked out a gene, we didn't get a disease, this is not such a surprise. But it tells us that what we think deterministically about these genes ain't quite so. Just today, we heard from the Laboratory of Molecular Medicine from Heidi Rehm. In one disease alone, in hypertrophic cardiomyopathy, they have shifted 71 variants from as more benign from more serious. Things change. These things were more worrisome, now they're less worrisome. 66% of variants were only seen in one family. Do we actually know they were causal? Just because it happened doesn't mean it actually caused it. And so I'm going to argue that there's 4 components to falsely positive incidentalome. Wrong annotations, measurement error, wrong priors, or the wrong comparison group, and multiple comparisons, which is essentially the curse of individuality. For each of these 4, I'm going to tell you there are ways to address this, and I am very optimistic about our future of individualized medicine. But we really have to be honest with ourselves how we are going to address this. So, like many others, we have a pipeline we called Genome, where we've looked at a variety of genomes, and we see what everybody sees, which is if you look at any given genome, you'll see approximately 400 genes with rare, novel, non-synonymous mutations at highly conserved loci. You'll see that there are genes with homozygous mutations implicated in disease in about 55 of them. And it's unclear how many of them are real and how many of them are poor annotations. But we actually did a study, and a study with Mark Tong I published last year, a study where we looked at three databases, one with 2 versions, and we found that the error rate varied between 25% to 5%. So this is the best we have to offer in our databases. And the error rate is 25% to 5%. And these are our best databases. Now, this is going to be cleaned up because there's several companies, like the one that Mike Snyder is involved with, Personalis, that have a business plan that is to clean it up. They will be cleaned up. But it's going to take time. And until then, there's a lot of noise out there, a lot of wrong annotations. And by the way, one of the things we point out, not only were the annotations wrong because basically people used the wrong reference mutation, but sometimes there were very huge syntactic errors. There was this very nice study that appeared in Genome in Medicine, where they did whole genome sequencing, and they showed that you could create a phylogenetic tree. So looking at sequence similarity across the whole genome, and perhaps not surprisingly, the Koreans were near the Chinese, the whites were there, and the Africans were together. And what was interesting about these two Africans, not only were they together, they were the same person. So this is a riff along what Mike was telling us about, about different platforms giving you different sequences. And again, this too will be improved. It'll be improved because sequencing technology will improve, depth of reads will be improved, different use of different baits will be improved, and complementary technologies will be used. But where we are today, it's pretty much here. So even though every platform will tell you we do 99.9% accuracy or better, those two Africans who are the same guy will disagree. So sequencing error, yes, it's exactly what I said. And the point is, we have to remember, we're looking at big numbers. So even if we take at face value, which I challenge you whether we should or not, out of a billion bases, even those kinds of very good error rates will still give you between 1,000 and 100,000 errors. So in the very little part of my life that now, one clinic a month that I actually take care of patients, I don't want to be reporting on those errors. And these are chemistry dependent, and as I said, it will get fixed eventually. I'm quite confident about it. There's a lot of money involved. People will make sure this happens. And then there's the wrong prior. So when I show something like prior probability, everybody is going to fall asleep, you think, because it sounds like statistics. But really, I'm going to make it very, very biological, because this is really a question about which group are you the most like? Which group is your genomics making you the most like? So this is one of my favorite stories. So it's well known in the literature that this disease called hemochromatosis, a storage disease where you basically, you accumulate iron in all your tissues, and it kills your endocrine glands, it destroys your heart. It's a bad disease. And if you look at a hereditary hemochromatosis clinic, 80% of the individuals in the hereditary hemochromatosis clinic will be homozygous for a mutation in HFE. So internalize that, HFE gene, hereditary hemochromatosis clinic, 80% of them will have a mutation in that gene. So some very smart people decided to sequentially genotype 41,000 individuals for those mutations found in the hereditary hemochromatosis clinic for a set of variants that are textbook highly penetrant mutations. And guess what they found? What they found was that, sure enough, 151 individuals in this 41,000 were homozygous for this mutation. But of the 151, only one out of the 151, one had any clinical, biochemical, or family history evidence of hemochromatosis. So this went from a highly penetrant Mendelian disorder to a common variant with weak penetrance. How did this happen? It happened because what is shared in the families where they actually ascertain these mutations? What's shared? What? Thank you very much. So your co-ancestry, so you have shared genetic background. What else is shared? What? Environment. Thank you very much. Environment. And if you don't share both the environment and the genetic background, all bets are off. And in fact, we've seen this in many other diseases. BRCA1, which we heard about today, the penetrance of BRCA1 mutations, first ascertained as being very high in Ashkenazi families with breast with a family history of breast cancer, the penetrance of BRCA1 mutations has decreased in every single year since the first publication. Are we told this? No, but it's there in the books. And by the way, we already knew this, because if you look at mouse models of hemochromatosis, these are all mouse models of hemochromatosis. Same mutations, different strains. Only in 1 out of 3 strains do you see the pileup of iron that you see in hemochromatosis. So these are the same species. The only difference is genetic background, which is, by the way, why so many mouse-borne genetic diseases never make it to be relevant in human beings. Because in the end, we're highly redundant organisms. We don't want to fall over at the slightest breath. And so it takes unusual combinations of genetic background and environment to affect us. And by the way, here's a sad story. It turns out the more alcohol you drink, this is amusingly a French study, the more alcohol you drink, more penetrant. So it's one of the bad stories for alcohol. The more you drink, the more you are set up for higher penetrance for it. So we've got to understand the genetic background, which whole genome sequencing will allow us, and we've got to understand the environment. And given the fact that most of our diseases, common diseases, have heritability around 50%, that tells us that 50% of the variance is from the environment. So we better start sharpening our knives and looking elsewhere as well. So I think we're addressing the wrong priors looking at these case studies. We need to get big population studies. So this is a vote in favor of large numbers of patients. We do need the billion genomes that Mike Snoder talked about. But until then, we actually need real doctors with clinical ascertainment. What do I mean by that? It's when a doctor actually thinks. Now, when I say a doctor, I mean a doctor that we almost don't have anymore, thinks that you have a disease. That's a good time to have a genetic test, because remember I told you that most genetic tests have 80% specificity and sensitivity? But if you have, for instance, a family history of hemochromatosis, or if I see that on your routine cell blood count evidence of an iron deposition disease, then my prior probability is not 1 in 5,000 that you have hemochromatosis, it's 1 in 10 or 1 in 5. And so now, even a poorly performing genetic test works great. So you have to use priors. And if we don't have that, then if all diseases have equal priors, you are setting yourself up for incidental omicity, just as you would be if you received, as it used to be offered for people's birthdays, total body MRIs. You're going to find something. So, by the way, so how do we get these large populations? So let me just take a little detour. I was awarded 8 years ago a $20 million grant that's been since renewed for creating a National Center for Biomedical Computing called I2B2, and where we said we were going to use the informational byproducts of healthcare delivery to actually measure things about patients and to actually get patient samples. And this toolkit called I2B2 is what I'm going to talk to you about in a few slides. By the way, I just want to look at the organizers. According to this, I have been only talking for about 22 minutes. On the other hand, we're kind of running late, so I'll try to hurry up a bit. So this toolkit that we've developed, and I'll tell you more about it, has actually multiple software components, and you should not care about these multiple software components except to know that it's free and it's open source, and it's been adopted by 60 academic health centers across the United States. 60 academic health centers, major ones, and this is out of date now, 12 academic health centers across the world, because everybody's trying to understand what's going on in their patient populations and how to study them. And the typical toolkit provides is the ability to look for any set of patients at both the data that came out of the text. We use natural language processing techniques, computational techniques, to take the text that doctors use to talk to one another, to codify it, as well as the codified data in the electronic medical record to put it together. And why do I make this point at all? Just because the billing data, the diagnostic codes, to use the vernacular, suck. They're actually maximized for income, and therefore they're not accurate, they're biased, and they're also not fine-grained. So by using both the narrative notes and the codified data, we're able to really get very fine-grained phenotypes, And we've shown in multiple occasions and multiple publications that we can, for example, repeat a genome-wide association study, in this case on rheumatoid arthritis, and shown in blue are the previous studies, in red the study that we did, and this study was done literally for 1% of the cost and 1/10 of the time of the previous studies using the healthcare record and discarded samples from our patients. And not only were we able to do this study fast, but we were able to repeat it for studies that had previously not been done. For example, anti-citrulline protein antibody is a standard clinical antibody used that gives you high specificity for rheumatoid arthritis. But what about if you have rheumatoid arthritis without being ACPA positive? Well, those GWAS studies don't exist, but we were able to just flip a switch on our electronic medical record, get those patients, and we could run it again for ACPA negative patients. Moreover, who are underrepresented in large genetic cohorts? Underrepresented minorities, Native Americans, I guess you don't call them Native Canadians, and African Americans, African Canadians, and so on. But it turns out, perhaps for the wrong reasons, but nonetheless it is true, that those same groups are overrepresented in tertiary healthcare centers. And so we were able to run very fast, it's the same study on African Americans and Hispanics, and show that actually in this particular instance, their odds ratios were very similar for the same SNPs. So we're able to address a disparities need that was not previously shown. But if you really want to revolutionize healthcare and look at personalized medicine, you really have to understand where we are today. And so, for example, through using I2B2, we did a study, this is 1997 to 2006, where of just two hospitals, MGH, Massachusetts General Hospital, and the Brigham Women's, two very good hospitals. And this is the baseline for heart attacks. And this is a huge mountain of heart attacks, that we flew our patients into with an 18% increase in heart attacks, adjusted for all the confounders you can imagine. Anybody have any idea what caused this mountain? Not you, Roden.
9/11.
What?
9/11.
I heard new drug. Vioxx. And when we case controlled it on the actual individuals, it was Vioxx. So think about it. First thing, we did not know we were killing our patients. We had a 18% increase in heart attacks, and we did not know it. You know what? You're doing it to your patients here with other drugs, and you don't know it either. So we're not looking at our own healthcare center. And it's not an inhuman possibility, because guess what? Kaiser Permanente, after the first publications and a bit of data, Kaiser Permanente took it off their formulary. They saved their patients. Nobody else did. We're talking about big effects here, not some of the subtle odds ratios of 2 to 3. Big effects. We completely neglected our patients because we don't look at our primary data. Now you could say, Zach, that's a nice party trick. You did this after the Vioxx disaster. Fine, I'll accept that criticism. But then there were two front page articles in the New York Times, above the fold, about a drug called rosiglitazone, one of the ones that Mike said he might have to use, rosiglitazone. And they were front page articles. Why? Because in one case, there was a congressional hearing that SmithKline Beecham may have sat on the data that there was a higher incidence of heart attacks with rosiglitazone relative to other drugs in the same class for treatment of diabetes. And then the second was because the FDA had some hearings about the same drug. Okay, drug companies lie. What's the deal here? The deal is we had published 9 months before any of those headlines on our own patients showing in diabetes care, showing that rosiglitazone versus pioglitazone, drug in the exact same class, had a statistically significant increased risk to our patients, that we were hurting our patients. Why weren't we doing this all the time? We actually did this in the heat of battle, and we were consequent. But if we're going to be so smart about all these customized drugs based on genetics, if we can't do this, how are we going to do it? The data's there. This is another study that was done by one of my faculty, Ben Rice, appeared in Science Translational Medicine a few months ago. and they simply took off-the-shelf data that you have all access to, drug safety data, taxonomic data about how drugs relate to one another, and just molecular characteristics of the data of the drugs and their targets in the public domain. And they trained up a network, a simple network, to predict adverse events based on a training set of data from 2000 to 2005. Then they said, can we predict new adverse events from 2005 to 2010? And they predicted that network going from all those drugs to all those adverse events with 80% accuracy. How come we're not doing this at the FDA? How come we're not doing this in our own healthcare systems? These are not complex analyses. And although I think we need to push ahead full speed with the genomics because it will inform this, our game is actually not good enough today to actually use it. And by the way, quick aside, there was a study done in 2003 of primary care physicians, and they were asked, have you ordered a genetic test for cancer susceptibility in the prior year? What percentage of those doctors do you think said, I had ordered a test? 5%? Good. If no one else says anything, your average is now better than the average of about 400 audiences I've asked this of, because most audiences say 1% and the actual answer is 30%. And that's always surprising to people. And then I ask them, what do you think was the best predictor of the doctor asking for ordering that test? And the answer is the patient asking for the test. And because they have a family history, they Google the disease, they say this is BRCA1 test, and they ask the doctor. Similar studies, other studies show that doctors are A, uncomfortable, B, incompetent to interpret these tests, and C, when they're detailed by attractive men and women from the drug company, from the lab testing company, in 4 cities, as reported by the CDC, the ordering of the same test that they're uncomfortable with and incompetent in interpreting goes up by a factor of 4. So that's why I put real doctors in quotes before. So we need to be able to look at this in a larger fashion. So this I2B2 system I just described is now, as I told you, had wide adoption. And 5 of the hospitals that we have at Harvard that are affiliated with Harvard have adopted I2B2 independently. And I'm going to tell you that these hospitals probably get along worse than even you get along with your neighbors across the street. And, but nonetheless, we were able to agree, without creating a centralized database, could we actually create a distributed sharing network where we share aggregate data in real time without actually creating a shared database? And the short answer is we did it, and this represents 6 million patients and 10 billion facts. And for example, in 1 month, I was able to do a study here of autism where we had 14,000 patients. Is that right? Yeah, 14,000 patients. And I was able to do a comorbidity study of autism where the previous biggest study was of 200 patients. And I did this single-handedly, of course, using shoulders of giants that had actually implemented all the software. And I was able to show, for example, of course, that there's an increased risk of epilepsy, as well known, and that there's not only an increased risk of bowel disorders, but of actually bowel disorder-related procedures. But I was also able to show that inflammatory bowel disease was actually very significantly elevated compared to the general population. And now, of course, Mark Daly, who's one of my colleagues, who's interested in both autism and IBD, is now looking at their joint appearance. But again, these are simple to find facts, or we are not empowered or not willing to look at our healthcare system to find these data. And that's going to be a huge impediment to forward motion. We're using the same I2B2 network I just showed you this year to actually create an 8 academic health center network to do these autism comorbidity studies and type 2 diabetes studies using this essentially volunteer organically grown network. So now, that was my long-winded answer of how we're going to get rid of the second problem of the wrong priors. Let's get big data on large numbers of patients. But then there's a problem of multiple comparisons. And so with one variant, picking the right comparison group is feasible. But if you only focused on 10,000 important variants, and each of them with a 1% frequency, you might be able to find a large enough group of human beings to study it. But with two variants, the chance of those two variants co-occurring is small, and with three variants, even smaller. So we're not with all the pairs of 10 to the 4 variants, we don't have enough people on the planet to be able to do the right statistics. It's just not going to work. So what are we going to do? We're going to have to, when you run out of people, you're going to have to start going back to basic physiology and study the interaction, as the Cancer Genome Atlas Project is, across multiple modalities. And I think the best example of this is iSnyder, which is looking over time, physiological and various proxies of physiology across multiple modalities as a discovery process, not as a clinical delivery process. And I think he is showing us the way. And I think this will allow us to have the beginnings of mechanistic interpretations, because population studies alone will be insufficient. So the challenge is the one I showed you before, to do that pipeline and to bring back that pipeline so that you can understand at every point in pipeline how you arrived at each of the conclusions, and to update it in real time. And this is no mean feat, with news coming from the whole combined life science output of our world. And I just want to put a quick plug that we just closed the admission to our contest that we're having at Harvard, called the Clarity Contest, where we're putting several family genomes out for the contestants to interpret, and then we're going to see how well they do. And so stay tuned. We'll hear more about that in late fall. So there was a very interesting article that appeared in Science Translational Medicine by Roberts and Vogelstein, basically the Hopkins Mafia. And it was a very interesting article, because based on a study of large twin studied cohorts, looking at identical twins and non-identical twins, they were able to come up with very, very good estimates of heritability for on the order of 18 different common diseases. Moreover, they also have the frequency of the diseases. So they were able to show for what percentage of these various disease populations would you be able to have, say, something informative to the patient if you could interpret fully, not one variant, but the whole genome, full interpretation of the whole genome. And this article, which generated a lot of controversy, and I invite you to look at the responses to their paper and then their response to the responses, which is extremely entertaining. But in this controversial paper, they point out that for most diseases, there's not going to be much information for a given patient. But most patients, it'll be informative for one disease. So there's a cup half empty, half full. And by the way, most of the information seems to be coming around cardiovascular disease. So in this study, what they showed was in, let's be clear, and they didn't make it that clear, and in a commentary I made it very clear, this is for use of genomes for asymptomatic individuals. For asymptomatic individuals, for most diseases with perfect genomic interpretation, it's not informative. But for every individual, it'll be informative for at least one disease. So it's cup half full, half empty. And in order to address this, I wrote a paper, well, I was thinking about this long ago, and I wrote a policy piece in Science in 2007, where I talked about how to use healthcare systems to actually start doing research, but allowing research to be factored in to clinical care and vice versa. Because I believe, and this is too very controversial, that when we learn something about patients in research, we have to be able to translate it into care. And to give you the icon for that, there was a study of prenatal screening for Tay-Sachs disease, in which consent did not include disclosure to the parents if they found trisomy 21, Down syndrome. Is that right or wrong? If you're doing a study of gestational risk for Tay-Sachs based on genetic assessment in utero, is it right to not disclose to the parents whether there is trisomy 21. I'll leave that, and I can read the paper on your own time. I asked you, what is the genome worth? Here's where it's worth a lot. This is the work done out of University of Wisconsin, where they looked at a poor child who had hundreds of operations for this unspecified bowel disease, and based on whole genome sequencing, they finally were pushed over the edge to do the bone marrow transplant, the stem cell transplant, which actually caused essentially all the symptoms to go away. That's a great story, but I want to point out, it's not an asymptomatic individual. It's an individual with an actual present chief complaint. It's much more of the clinical context. Which leads me to, and let me assure my organizers I'll be done in 3 minutes. A report that I was privileged to be part of from the National Academy of Sciences called Toward Precision Medicine. This is freely available online from National Academy Press, where we talk about how we're going to create, just as Google has layered multiple modalities on top of satellite imagery to create the whole ecosystem that you know well, we should be creating an information commons across all the exposures from the environment through the genome on down to individual patients to actually understand the interactions of these various parts, so that not only do we go from the standard basic science to biomedical research pendulum, but we extend it to observational studies during the course of clinical care, using our electronic health records, using environmental data, so that we can actually, and using molecular data, so that we can actually have not only a GPS to tell about the harm we might be doing or the opportunity we have to treat our various patient populations, but to provide patients their own GPS so they can also individualize their care. This is realizable, but it's only realizable if we have enough data and we analyze it thoughtfully. And with that, I'm going to end my talk as follows. The threat of the incidentalome is acute, it's present, and it's our problem. No one else is going to solve it for us, and it threatens the translation of genomics. If we do this willy-nilly, there'll be so many bad stories of people removing parts of their body for no particular gain that there will be, in fact, a pushback. And many of the components I predict are going to be addressed in the near term, so we should be optimistic, but we should make that near term happen, and we'll have to go both big studies and small in terms of systems biology, and we need to understand context much, much better and create knowledge-sensitive doctors, or else we'll be in trouble. And with that, I want to thank you and my collaborators.
Maybe one or two questions. But
Thanks for a great talk. You've made it very clear what the pitfalls would be in using genomic data to assess risk of disease and then act on that. What about the use of genomic data for in the service of preventing drug toxicity? You can combine that with large hospital databases and use drugs much more safely.
I think that's one of the near-term wins. And I think that we have much more of the data, and I think we're much more solid ground. I think we start getting on weaker ground when we start looking at some of the interesting variants in pseudogenes and others for supposed metabolizers that are interesting but are not yet validated as informing us. But for where we already know, where the PK/PD people have already told us these are important metabolizer genes, I think we're in very good shape to use that to prevent bad things happening to our patients.
So Zach, how much money did this cost you, and what type of infrastructure did you have to put in place?
Okay, so the infrastructure, the I2B2 infrastructure, obviously it was a lot of money from NIH, but I can tell you now, because other academic health centers have adopted it, and it's not cheap. Even though our software is free and open source, there are other companies, I wish I had a cent on their dollar that they spent, but there are other companies that have actually taken our software and have implemented it at these large academic health centers, and it does take $2 to $3 million to get this up and running. So it's not free, and it also, by the way, obviously requires a change in mindset of the leadership of your hospital and your healthcare system, that you're actually going to look at your patients and you're going to do something if you find something, and that's not always a foregone conclusion. So thank you very much and enjoy lunch.